Hi everyone, welcome to Chapter 5, Protein Function. In this chapter, we will explore how proteins interact with other molecules and how these interactions are related to dynamic protein structure. In this lecture, we will understand as to how proteins interact with other molecules. The learning goals of this chapter are as follows. We will first look at methods of binding ligands and proteins and understand how protein-ligand interaction works. To examine this further, we will look at quantitative and graphical modeling of protein-ligand interactions. And then we will look at some examples. We will look at interactions of globins with oxygen and non-oxygen ligands. Furthermore, we will look at physiological regulation of oxygen binding. And finally, we will look at three different mechanisms, beginning with the mechanism and control of antigen-antibody interactions. Second, we will examine the mechanism of muscle contraction and regulation of muscle contraction. In Chapter 4, we looked at structures of globular proteins. In this chapter, we will examine certain functions of globular proteins. To be specific, one of the characteristic cellular function of globular proteins is to bind molecules. And a molecule bound reversibly by a protein is called as a ligand. Now, when a protein binds a ligand, there is always specificity involved, which means that the site where a ligand binds in a protein, which is called as a binding site, is almost always specific for a protein. For example, if I were to draw a structure of a protein, let's assume that this is a protein. A ligand can bind in this site, and so this is termed as the ligand binding site. Now, ligand binding is often coupled to conformational changes, sometimes quite dramatically. This means that once a ligand binds a protein, it can alter the structure of this protein, which means that once the ligand binds, the structure of the protein can change like that. That depends on the nature of the ligand and the site where it binds a protein. In multi-subunit proteins, conformational changes in one subunit can affect the others, which means that if you have a protein with multiple subunits, for example, if there is a protein that has 4 subunits, 1, 2, 3, and 4. Now, in this case, if a ligand were to bind in this site, this means that binding here can alter the properties of a site distant from this specific subunit, and this can make another ligand bind here, and that is how ligands can affect one subunit, or binding of one subunit affecting the binding of another subunit to bind another ligand. And this is termed as cooperativity, and we'll learn about this. Interactions can be regulated. This is illustrated by proteins like hemoglobin, various different antibodies, and proteins that control muscular movements. There are a variety of functions that are associated with globular proteins. The first one is storage of ions and molecules. And this is done by proteins called myoglobin. We have seen this before. Ferritin. Transport of ions and molecules. Hemoglobin is a characteristic protein that transports oxygen in blood. Serotonin transporter is another example. Defense against pathogens. Antibodies are one of the best examples that can that can initiate defense against a foreign body like a pathogen. Cytokines, another example. Muscle contraction. Actin and myosin are two different proteins that work in tandem to initiate muscle contraction. Biological catalysis. Globular proteins can also be enzymes. Two characteristic enzymes are chymotrypsin and lysozyme. The interaction of proteins with other molecules is almost always reversible. Reversible transient process of chemical equilibrium is given by this equation. A molecule that binds to a protein is called a ligand. It's typically a small molecule. But small proteins can also be ligands of other proteins. And as I said before, a region in the protein where the ligand binds is called as the binding site. Ligands bind via same non-covalent interactions that dictate protein structure. It allows the interaction to be transient. So let us look at the quantitative description of binding. Consider a process in which a ligand binds reversibly to a site in a protein. For example, if P were to be a protein and L were to be a ligand, and L binds to P in this site. Now, this is dictated by two different rate constants. Now, what is a rate constant? Rate constant is what defines how fast a reaction happens, right? Now, in this case, the reaction is binding of protein P by ligand L. And when they interact, a complex called PL forms because P interacts with L. And how does P interact with L? That is exactly what we learned in chapter 3 and 4. Of course, a little bit in chapter 2 as well. Non-covalent interactions or weak interactions in proteins that are dictated by amino acid side chains, right? Could be hydrophobic, hydrogen bonding interactions, van der Waals interactions, or ionic interactions with the ligand. So, gives rise to PL and is dictated by two rate constants, right? This can be quantitatively described by an association rate constant or a dissociation rate constant. Association rate constant dictates how fast these two interact to form PL. A dissociation rate constant dictates as to how fast the PL complex dissociates to form P and L. Remember, from your knowledge of Chem 1 or Chem 2, kinetics is described by whether a reaction is first order, second order, or to a lesser extent, third order. It could be zeroth order as well. Now, this is a second order reaction. There are two reactants. So you have to understand that Ka has a different unit as compared to Kd because Kd dictates going from one reactant to two products, whereas Ka is two reactants to one product. This can sound confusing, but let us break this down. So, after some time, the process reaches equilibrium, where the association and dissociation rates are equal. So, by law of mass action, you can consider Ka times P times L is equal to Kd times PL. Now, there's another way as to one can look at it. Is look at the equilibrium constant, big Ka. Remember, this is small Ka and small Kd. Big Ka is the equilibrium constant that dictates this reaction or this interaction. And big Ka is equal to the concentration of PL divided by concentration of P times concentration of L. This can also be equal to the ratio of Ka, small ka, and small kd. So the equilibrium constant depends on the individual rate constants. So that is how we break down the rate constant. Remember, the individual rate constant depends on the equilibrium constant. We can also express the equilibrium constant Ka as a reciprocal of Kd. So Ka is equal to 1 over Kd. Ka is obviously the equilibrium constant for the forward reaction, and Kd the equilibrium constant for the reverse reaction. And remember, the equilibrium constant is characterized, equilibrium composition is characterized by the equilibrium associate constant Ka and the equilibrium dissociate constant Kd. And this is different from small ka and small kd. So you have to understand the difference between large Ka, large Kd, and small ka and small kd. Large Ka is equal to ratio of small k over small kd, and this is a useful relationship that you have to remember. To understand the binding of a ligand to a protein, it is important to analyze in terms of the bound fraction. If we consider this equilibrium constant equation and rearrange it, we see that the ratio of bound protein to the free protein is directly proportional to the concentration of the free ligand. When the concentration of the ligand is much higher than the concentration of ligand binding sites, the binding of the ligand by the protein does not appreciably change the concentration of free ligand. That is, L is a constant. Let me explain to you what this means. If you have a protein and if you have a ligand, right? Now, what it says here is the concentration of the ligand is much higher than ligand binding sites. And in this case, the binding of the protein to the ligand does not appreciably change the concentration of the ligand. Here is the bound protein. And here are other ligands. So, to start with, ligand concentration is much higher than the concentration of ligand binding site. And that's the ligand binding site here. So let's say, call it ligand binding site. Now, this is much, so concentration of ligands is much higher than concentration of ligand binding site. In that case, protein binding to the ligand will not change the concentration of the ligand. This is usually the case in cells because the concentration of ligand is much higher than the concentration of protein. So we need to analyze in terms of bound fraction here. We can now consider the binding equilibrium from the standpoint of the fraction y, that is the ligand binding sites on protein that are occupied by a ligand. Y is equal to binding sites occupied by total binding sites. That is given by the term theta, which is equal to PL divided by PL plus P. And theta is a fraction of occupied binding sites. So how did we arrive at this equation? Now, if I break it down for you, now let's say we have 5 proteins in solution in a test tube. Now, all 5 proteins have these 5 binding sites, right? This is the binding sites of these proteins. Now, if 2 of these binding sites are occupied and 3 are not occupied, and the question is, what is the fraction of the binding sites that are occupied? And that fraction is this, right? Number of binding sites occupied is 2 divided by 2 plus 3, the total. And 3 is the number of proteins that does not have its binding site occupied by the ligand. So this is the fraction. That is what is given by this equation here. So in practice, we often determine the fraction of occupied binding sites, and it is given by this equation. Now, we need to determine how many of them are occupied. So PL is the one that needs to be calculated. We can use this equation, the equilibrium constant equation, to substitute for PL. Substituting PL with Ka times concentration of L and concentration of P will eliminate PL, right? We take these two terms to the left hand side and you substitute for PL in this equation. So Ka times L times P divided by Ka times L P plus P. Now in doing so, you are eliminating P and rearranging the equation, resulting in this specific equation in terms of Ka. So you can rearrange this equation and you can get this equation. And so, in terms of the most commonly used equilibrium dissociation constant, theta, or fraction of occupied binding sites, is given by the ratio of concentration of ligand to concentration of ligand plus Kd. The binding of a ligand to a protein can be analyzed graphically. The fraction of bound sites depend on the free ligand concentration and Kd, or 1 over Ka. The equation is this. Experimentally, ligand concentration is known because when you do an experiment, you always know the concentration of protein and the concentration of ligand. What you need to determine is how much ligand binds to a protein, and that is directly related to how much of the protein ligand binding site is occupied. Kd can be determined graphically or via least square regression. Now, an equation of the form x is equal to y over y plus z describes a hyperbola. And so, y is thus found to be a hyperbola, and it's a hyperbolic function of L. The fraction of ligand binding site occupied approaches a saturation asymptotically as ligand concentration increases. Again, the ligand concentration at which half of the available ligand binding sites are occupied corresponds to a Kd, or 1 over Ka. Now Kd is the more commonly used equilibrium constant, or it is called as a dissociation constant, because it is intuitively simpler. This is another example showing a plot of theta versus L. When L equals Kd, half of the ligand binding sites are occupied, as you can see from the plot here. As L falls below Kd, progressively less of the protein has ligand bound to it. For 90% of the available ligand binding sites to be occupied, L must be 9 times its Kd. And 9 times 5 is 45, which is here. And that is when you will have 90% of available ligand binding sites occupied. In practice, Kd is a much more applicable unit than Ka. Much more applicable equilibrium constant. Now the mathematics can be reduced to simple statements. Kd is equivalent to the molar concentration of ligand at which half of the available ligand binding sites are occupied. At this point, the protein is said to have reached half saturation. So this is only half saturation, which means half of the available ligand binding sites are occupied. The more tightly a protein binds a ligand, the lower the concentration of a ligand required for half the binding sites to be occupied. And thus, the lower the value of Kd, the better the affinity of a ligand towards a protein. Now if you consider another ligand which has a plot like this, and if you find the Kd from this, Kd is about 25 nanomolar, right? Now this is 5 and this is 25. This means that this ligand has a lesser affinity for the protein as compared to the first ligand. This has a higher affinity. The binding of a ligand to a protein can also be expressed as a semi-log plot, wherein the concentration of ligand can be expressed in logarithmic scale and theta as a fraction. And you will get a sigmoidal kind of a plot wherein the Kd is obtained again as the ligand concentration where half of the binding sites of protein are occupied. Let us consider some thermodynamic terms with respect to the binding of a ligand to a protein. Interaction strength can be expressed as association constant, which has a unit of per molar, or a convenient term Kd, which we talked about in the previous slide, which has units of molar. Kd, as you know, is 1 over Ka. Interaction or binding free energy is usually delta G0, or the standard free energy, which is units of kilojoules per mole or kilocals per mole. Now you know all the definitions, right? Delta G0 is equal to delta H0 minus T delta S0, enthalpy and entropy. And you also have the equations for Ka and Kd. The relationship delta G0 is equal to minus RT ln Ka, or RT ln Kd. Remember, when you invert Ka, the negative sign goes away. So delta G0 is equal to RT ln Kd. The magnitudes, as I said before, the lower the Kd, the stronger the binding of a ligand to a protein. As the Kd increases, the binding becomes weaker. The color bars indicate the range of dissociation constants typical of various classes of interactions in biological systems. A few interactions, such as between the protein avidin and the enzyme cofactor biotin, fall outside the normal ranges. The avidin-biotin interaction is so tight that it may be considered irreversible. It has a binding affinity of 10 to the minus 15 molar. That is extremely tight. Sequence-specific protein-DNA interactions reflect proteins that bind to a particular sequence of nucleotides in DNA, as opposed to general binding site to any DNA site. And that has a range of Kd between 10 to the minus 11 to 10 to the minus 7 molar. So if you get to 10 to the minus 12, that is picomolar, 10 to the minus 9 is nanomolar, and 10 to the minus 6 is micromolar. The enzyme substrate interactions is typically till low micromolar to low millimolar, whereas antibody antigen interaction is between picomolar to nanomolar. So it's a very tight interaction. There are two different models that dictate the specificity of a ligand towards a protein. The first one is called as a lock and key model. In this model, proteins typically have high specificity and only certain ligands bind. High specificity can be explained by the complementarity of the binding site and the ligand. Complementarity in size, shape, charge, or hydrophobic and hydrophilic character of proteins. The lock and key model was first proposed by Emil Fischer, and it assumes that the complementary surfaces are preformed, which means that a protein has evolved with a specific complementary surface for a specific ligand. Now, this is a typical example, right? Now, in this case, protein has a preformed complementary surface for a ligand which can bind to that specific surface. And this is the lock and key model that is proposed by Emil Fischer. The second model is called as the Induced Fit model. And this model is proposed by Daniel Koshland, and this assumes conformational changes on a protein. And these changes may occur upon ligand binding. This adaptation is called induced fit. This means that when a ligand binds to a protein, it induces a conformational change. Induced fit allows for tighter binding of the ligand. And induced fit allows for high affinity for different ligands. Both the ligand and the protein can change their conformations. Now, this is an example. And to start with, the protein does not have any preformed surfaces, or it does not have any complementarity towards the ligand. But when the ligand gets closer and it binds, it changes the conformation of the protein and also of itself. This is the very basis of induced fit.